So this morning, I'd like to tell you about our chemical probe we developed for Chikungunya NSP cysteine protease. This has been part of the READDI-AC program, one of the AViD centers. Alpha viruses are enveloped spherical, positively stranded RNA viruses. They're vector-borne, usually by mosquitoes, and there's worldwide outbreaks which result in arthritogenic or encephalitic disease. And you can see here listed the various virus classes and their diseases and the locations in which these diseases are found. Shown in the bottom right are the outbreaks of EV from 2006 to 22, showing a small but considerable number of outbreaks in the US, and the mortality rate is between 30 and 50% for triple EV. The alpha virus, specifically the Chikungunya replication complex, consists of a polyprotein, which is broken into several individual proteins by the action of NSP2 protease. So the polyprotein shown here is snipped in 3 different positions to give the constituent NSP1, NSP2 helicase, NSP2 protease, NSP3 macrodomain, and NSP4 RNA-dependent RNA polymerase. And these proteins form the basis of the replication complex. NSP2 protease is a papain-like cysteine protease, and I've shown the catalytic cycle here, which basically involves the action of a cysteine self-hydryl group on the amide bond to be cleaved. And we started our investigation of inhibitors of NSP2 protease by screening a library of about 6,000 covalent compounds, which is commercially available. As you can see here, this library contains different covalent warheads, and out of the about 6,000 compounds that we screened, the warheads containing vinyl sulfones was the warhead which showed up most consistently in this screen. So a potent vinyl sulfone inhibitor was identified, RA2034, and this showed IC50 of 60 nanomole in the protease in vitro assay. Obviously, it's potent against the enzyme in an in vitro assay, and it's also potent in and has nanomolar potency in an antiviral assay using a reporter. Well, during our investigation of 2034, we found that the compound did undergo spontaneous cyclization to this bicycle shown here. And we showed that this compound, number 2, was totally inactive with an IC50 above 40 micromole. So after 48 hours and phosphate buffer around pH 7, 20% of 2 was present. So we realized that this was a significant problem for this series of compounds. And since we've published this work, commercial samples of the acyclic potent inhibitor often are contaminated with various levels of compound number 2, which obviously reduces the activity. We set out to explore the SAR of the 3 different portions of this inhibitor, firstly by making over 100 analogs where we changed this portion of the molecule, looking at different substitutions, different heterocycles, replacing the ethoxyphenyl group. We also looked at 20 different warheads, changing the vinyl sulfone to various warheads, and also looked at 15 unique heterocycles joining these 2 portions. And the compound we ended up with was a compound where we replaced the isosteric pyrazole with an isoxazole. And this compound was almost as potent as the original 2034, having an IC50 of around 40 nanomole, And more importantly, this compound, 3022, was now stable at least after 48 hours at pH 12. And the traces here show that there's no evidence of the cyclization, which is not possible now. We've replaced one of the pyrrozole NHs with an oxygen. So the properties of the compound, which is now known as SGC NSP21, are shown here. Medium molecular weight, good log P, pretty potent ligand deficiency. No pains alerts, like I just shown, it's stable at room temperature between pH of 3 to 12. Good aqueous solubility relative to its potency, and no evidence of aggregation by dynamic light scattering at 100 micromole. But the biological profile is obviously that it targets Chikungunya NSP2 protease. The in vitro potency was 40 nanomole, and we have shown some SAR, which is good, showing that there is some definite SAR around this molecule. In a minute, I'll show you the target engagement studies, but we know that it targets cysteine 478 in NSP2 Pro, and as it's a covalent inhibitor, measuring an IC50 is not strictly the best way to measure its potency. We need to measure the Kinact over Ki, and this was measured at 6,000 per mole per second, which is quite respectable for a covalent inhibitor. I'll also show in a couple of slides that we have 30-fold selectivity over 23 human and viral cysteine proteases, and we have a good half-life against glutathione reactivity. In a couple of slides, I'll also show through TAMRA labeling and biotin strep pull-down that we have proteomic selectivity of binding as well. And in the antiviral assay, where we use Chikungunya end-look readout, we have an EC50 of 50 nanomole. And we've also checked the viral titer in other viral assays to make sure that the end-look readout does actually is a good replacement for the viral titer readout. And finally, we see no cellular toxicity up to 10 micromole. So SGC NSP21 inhibited the NSP2 protease in a time-dependent manner, as you'd expect for a covalent inhibitor. And shown here is the Kinact and the Ki, which results in the ratio of 6,000 per mole per second. We determined that this molecule reacts with the catalytic cysteine 478 of the NSP2 protease by incubating the compound with protein for 30 minutes, followed by a trypsin digest and identification of the fragments by LC-MS. And we clearly see this fragment showing up with the increased molecular weight, which corresponds to the molecular weight of SGC NSP21. So SGC NSP21 has pan-antiviral activity. If you look at the phylogeny of togaviruses, there are New World viruses such as EEV and VIV, and there are Old World alpha viruses such as the Chikungunya and Majorano. And you can see here where we've actually looked at the viral titer with increasing doses of SGC NSP21, that we have a dose-dependent 5 to 9 log decrease in the viral titer against infectious isolates of these three viruses. So we see pan-antiviral, anti-alpha viral activity with this compound. Looking at the cysteine protease selectivity of the chemical probe, we looked at various human enzymes, the cathepsins family, the USPs, and a bunch of other proteins which are known to be cysteine proteases. And you can see in the black lines that we see little to none inhibition of any of these proteases at 10 micromole treatment. The lines shown in gray are the corresponding activities in the presence of the known inhibitors for each of these enzymes, showing that we can inhibit them with their specific inhibitors. So we can say that we have at least a 30-fold selectivity for Chikungunya NSP2 Pro over other cysteine proteases. So we also wanted to show target engagement. So we made this compound here with an acetylene in the to replace the ethoxy group, and we incubated this with protein and performed a click reaction with the TAMRA azide. And when we did the SDS gel and fluorescence imaging, we saw no pull down in the presence of HEC293 lysates, which was nice to see that we didn't see any human proteins pulled down. And obviously, when we spiked in NSP2, we did see pull down of the viral NSP2 protease. We had to spike it in because obviously there's no viral proteins in human HEC293 cells. We also looked at the chloroacetamide version of the vinyl sulfonamide, and you can see that both with the spiked, we saw some NSP2 pulled down as well as some other targets, but there were also other targets pulled down in the absence of the spiked NSP2. Well, one of the questions we wanted to answer is whether the vinyl sulfone alone is responsible for the activity. And to that end, we made this compound named VSS, where we've replaced the ethoxyphenyl group with a propargyl group. And we did the same pull down studies. And again, we saw no reactivity with this compound, indicating that it is a characteristic of the complete compound with the ethoxy group present that's responsible for the activity. And finally, we made the biotin clicked reagent using the same propargyl compound that we made before. And when we did these pull downs, again, we see some non-specific binding in lanes 1 and 3. And where we spike in NSP2 protease, we see some NSP2 protease pulled down. Again, we did tryptic digest on lanes C3, and the LCMS was used to identify the enriched proteins. And shown here is a typical volcano plot of the proteins which were pulled down. NSP2 is highlighted here. It's the most pulled down protein. And there were 3 proteins, human proteins pulled down, which are not related to cysteine proteases. So no other cysteine proteases or other enzymes were identified as off targets. So finally, what are the aspects of a chemical probe is that it has a good negative control compound, which can be used in experiments. And to this end, we developed SGC NSP2-1N, which has an isopropyl group on the nitrogen. The key thing for a negative control for a covalent inhibitor is that the warhead is kept intact. Obviously, one of the other ways of making an inactive compound from this chemical probe would be to reduce the double bond, but that wouldn't be a true negative control because we would need to have the active warhead in place. So you can see that in the in vitro enzyme assay, SGC NSP2-1N is almost inactive compared with the chemical probe. And in the antiviral N-look readout, it's pretty dead compared to the chemical probe. Chemical storage is stable in solution at pH 3 to 12, as expected, just like the chemical probe. The synthesis of both of these compounds is fairly straightforward. This diketo ester is made from this diethyl oxalate, and cyclization using hydroxylamine gives the isoxazole acid. And then coupling with these two amines gives you either the probe, NSP2-1, or the negative control, NSP2-1N. So to summarize the characteristics of this compound, we have SAR for Ki, IC50, and K-inact for the non-covalent interactions. We do have a negative control available, which is inactive on both the antiviral and the in vitro assay. Solubility is good at around 20 micromole compared with the cellular IC50, and it is relatively stable in glutathione assay. The compound's stable at pH 3 to 12 for at least 48 hours. And we've shown proof of target engagement by indicating that it reacts with cysteine 478 to form a covalent adduct. And by TAMRA labeling and biotin pull-down, we've shown good proteome-wide selectivity using unbiased proteomics. We've also shown it's 30-fold times selective versus other cysteine proteases, and we have good activity in the antiviral assay with a K-inact over Ki of around 6,000 per mole per second. So we think we have a nice, stable chemical probe here, along with its cognate negative control. So in closing, I'd like to thank my colleagues at SGC UNC. Specifically, most of this work was carried out by Anirban. I'd also like to thank our UNC collaborators, specifically Mark Heisey and Nat Morman, who have been involved with the antiviral assays, and Ken Pierce, who performed the initial screening of the covalent set. And finally, I'd like to thank our other collaborators who have helped us characterize these molecules, as well as our funding sources, READDI-AC, NIH, and the SGC. So thanks for your attention. I'll be happy to answer any questions.